Welcome to the deep dive. Today, we're shrinking down, way down, into the microscopic world of plants.
That's right.
We're digging into some source material, showing how AI is being used to analyze, well, the hidden spaces inside leaves.
Exactly. We've got a consultation record and an image. And they basically lay out this research project tackling a really intricate problem,
No idea.
understanding the 3D arrangement of cells and those crucial air pockets within a plant leaf.
Okay. And
Yeah.
our mission for this deep dive is to unpack this specific project. Like, what are they trying to do?
Mm-hmm.
What are the challenges? Which sounds surprisingly tough, even with fancy tech.
Yeah, they are.
And what AI solutions are they actually proposing to measure and understand these internal structures?
It's a really great example, actually. It shows how this powerful tech, stuff you might think is just for like big data
Right.
is being applied right down at the fundamental biology level. It's about moving from just sort of seeing these things.
To actually measuring them.
Exactly. To quantifying them in a way that was just incredibly difficult, maybe impossible before.
Okay, let's jump right into the core challenge then. The heart of this project seems to be using AI to get precise measurements of two related things.
Yes, the stomatal complex on the leaf surface.
Okay.
And the empty space directly underneath it, the substomatal cavity.
Stomata, those are the pores for gas exchange, right?
CO2 in, oxygen and water out.
That's them, often with guard cells around them. And what's really key here is that right under that pore, typically there's this air-filled space.
The substomatal cavity.
The substomatal cavity, it's basically the gateway,
Right.
Connects the outside air coming through the stoma to all the air spaces deeper inside the leaf tissue.
So the goal isn't just finding the stoma on the surface. It's measuring both the stoma and this linked internal air cavity, automatically using AI.
Precisely. That's the first step. Use AI to automatically find them, mark them, measure their size, the stoma complex, and its corresponding cavity.
But the real scientific payoff, the thing they really want to get at, is analyzing the relationship between the size of that stoma opening and the size of its connected cavity.
Ah, so how they relate to each other.
Yeah, and then maybe classify those relationships, see if there are different types, different configurations.
That's interesting. And the data they're using, it's confocal microscopy images, the source says,
Correct.
using Arabidopsis thaliana, that standard lab plant.
Right, Arabidopsis. And confocal is key because it lets you capture optical sections, like slices, layer by layer.
So you can build up a 3D picture.
Exactly. You need that depth information. This isn't a flat 2D problem.
And the images use specific colors, fluorescent signals, to tell things apart. The source mentioned magenta for cell membranes.
Yep, magenta shows the cell outlines.
And green for the areas without cells, like these air cavities.
Right. Green means, well, likely empty space. An air cavity. That color separation is absolutely critical for the AI.
Because it tells the AI this is cell structure versus this is probably empty space.
Exactly. Magenta is cellular, green is non-cellular. The source even gives the specific wavelengths they use to get that clear separation, making sure the signals don't overlap and confuse things.
Okay, but here's where it gets, really interesting, the challenges they point out. Measuring these things automatically, especially the cavities in 3D, it's proving tricky.
Yeah.
What's the main hurdle?
The absolute primary one they highlight is just the incredibly complex 3D arrangement of the cells inside the leaf.
It's not neat and tidy.
Not at all. It's not like stacked boxes. You've got irregular cell shapes, tightly packed, interwoven in layers. It creates a really challenging, non-uniform environment for software to navigate.
And the tools they first thought of using, they aren't quite plug and play.
That's another challenge mentioned, something like StomaVision. It's apparently great for finding stomata in normal images.
Right.
But it might struggle with the specific characteristics of these confocal images, the contrast, the 3D nature. So using it means adapting it or maybe finding other tools for some steps.
Yeah. Okay, so adapting tools is one thing. What about actually mapping out that complex 3D structure you mentioned? How are they thinking of tackling that?
That's a core part of the plan. The source discusses using that magenta signal, the one outlining the cell membranes,
Okay.
to try and reconstruct the whole 3D cellular architecture of the leaf. They specifically talk about mapping the upper epidermis and the mesophyll cell layers underneath.
So like building a 3D scaffold based on where the cell walls are located?
Pretty much, yeah. And once they have that basic map, the idea is to go further and try to identify specific types of cells within those layers.
Like telling the difference between guard cells, pavement cells, mesophyll cells.
Exactly. Identifying stomatal lineage cells, guard cells, pavement cells in the top layer, and then the spongy mesophyll cells in the middle.
Okay, and how does having this detailed cell map help them with the green stuff, finding and measuring the cavities?
It gives context. It helps define where to look for the cavities and how to interpret the green signal. The source actually mentions two ways they might calculate the size of those green spaces.
Two approaches.
Yeah. One is pretty direct. Just calculate the volume of the connected green signal area, find the green blob, and measure it.
Okay. Seems straightforward. What's the other way?
The other is more indirect. It relies on that cell map we just talked about.
Right.
You could define a specific region, like a small cube of the 3D image, then calculate the volume taken up by the magenta-labeled cells within that cube.
Okay.
And then you subtract that cell volume from the total volume of the cube. What's left over should be the airspace volume.
I see. So one way measures the green space directly. The other calculates the space by figuring out how much isn't cells. That's clever.
It is.
But why? Why bother with the subtraction method if you can just measure the green signal? Is the direct green measurement maybe unreliable sometimes? Like noisy or patchy?
That's a great question. The source doesn't spell it out explicitly, but you've likely hit on the reason. Direct measurement of that green signal might be affected by, variations in signal strength. Maybe the dye didn't perfectly fill the whole cavity, or just general imaging noise.
Why?
Calculating by subtraction, based on the potentially more robust cell membrane signal, might give a more reliable estimate of the total available airspace in a region, even if the green signal inside it isn't perfect.
So they're exploring maybe complementary ways to get the most accurate numbers possible.
Seems likely. Hedging their bets, perhaps.
Okay, that makes sense. So given these challenges, the complex 3D stuff, adapting tools, mapping cells, what was the overall proposed AI toolkit? What workflow did the consultation suggest?
Well, the suggestions laid out a kind of multi-tool approach, not just one piece of software.
Right.
StomaVision was suggested for the first step locating the stomata, which maybe implies they think its core detection ability could be adapted or used as a starting point.
Before passing it off to other tools.
Yeah, perhaps. Then for finding the cavities, the green spaces.
What did they suggest for that?
That's where Cellpose comes in. Cellpose is known as a really good generalist segmentation tool. It's powerful for identifying individual objects in images.
Even though it was mainly designed for cells.
Exactly. But the source suggests it can be trained or maybe just configured to segment out the cellular regions, the magenta bits, from the non-cellular regions, the green bits.
So it can effectively draw the outlines of the cavities.
That seems to be the idea. Isolate the cavities by identifying everything else.
Okay, so StomaVision finds the stomata. Cellpose helps define the cavities. But this is all 3D. How do they actually build that 3D image and get the volume measurement?
But that's the job for ImageJ, or Fiji. Fiji is basically ImageJ with lots of useful plugins bundled in.
Yeah, ImageJ Fiji. That's like the workhorse of microscopy image analysis, right? Open source, widely used.
Absolutely. It's powerful enough to take all those 2D confocal slices.
Layers.
The layers, yes, and reconstruct them into a full 3D representation of the stoma and its connected cavity. And then, crucially, it has the tools built in to calculate the volume of those reconstructed 3D shapes.
So let me see if I've got the proposed workflow right. Step 1. Use StomaVision, maybe adapted, to find the stomata on the surface. Step 2. Use Cellpose on the 3D stack to distinguish the cells from the cavities, essentially outlining the cavities. Step 3. Use ImageJ Fiji to take those segmented layers and build the 3D structures of the stomata and their linked cavities.
Right, reconstruct them in 3D space.
Step 4, use ImageJ Fiji again to actually measure the sizes and volumes from that 3D data.
That's the core pipeline outlined in the source material, yes. Combining specialized AI for the tricky segmentation part with a robust platform for the 3D analysis and measurement.
Okay. And once they have all these measurements, size of stoma, size of cavity, for lots of examples, how do they plan to analyze that relationship between them?
For that, the source suggests a technique called Affinity Propagation.
Affinity Propagation? What's that?
It's a type of clustering algorithm, but instead of just grouping things based on, say, how close they are in distance.
Like k-means or something.
Sort of, but it works differently. It identifies representative examples or exemplars within the data by having data points send messages to each other about their suitability to be an exemplar.
Okay. So it finds natural groupings.
Yeah, it can help them group together the stoma cavity pairs that have similar size relationships or maybe similar structural characteristics. It lets them potentially identify distinct types of these stoma cavity units within the leaf.
Which could tell them something meaningful biologically.
Exactly.
This all leads to the bigger question, really. Why? Why is understanding the exact size and 3D shape of these tiny air spaces and how they connect to the stomata so important? What does it actually tell us?
That's the crucial part, the size and the architecture of that substomatal cavity directly impacts how efficiently gases diffuse.
Between the outside air and the inside of the leaf.
Precisely. Think of it like a little antechamber for CO2 going in and water vapor coming out. Its size, its shape, how well it connects to the surrounding mesophyll cells, all that influences the resistance to gas flow.
So it affects how easily the plant can breathe.
In essence, yes.
Right.
Being able to quantify this in 3D for many stomata lets researchers understand how different plants, or maybe the same plant under different conditions like drought,
Right.
how they might optimize this critical junction point for gas exchange. It directly links that microscopic structure to big physiological processes like photosynthesis and water loss, transpiration.
So understanding variations could explain things like drought tolerance or how plants adapt to high CO2.
Absolutely. It could shed light on adaptation to all sorts of environmental conditions, drought, heat, changing CO2 levels.
Okay, let's recap this fascinating deep dive into the micro world of leaves.
Sure. So we've seen how researchers are planning to combine some pretty powerful AI tools, things like StomaVision and Cellpose.
Right, the segmentation tools.
Right. With established image analysis software like ImageJ Fiji, and then statistical methods like Affinity Propagation clustering.
And they're bringing all this tech to bear on the really intricate 3D structure of plant leaves, specifically targeting the stomata on the surface and those hidden substomatal cavities just beneath them. Using confocal microscopy and those distinct fluorescent signals, magenta for cells, green for the spaces.
And the ultimate goal, remember, is to automatically measure and then classify the complex 3D relationships between these surface pores and the air spaces they connect to.
Moving beyond just looking to getting real quantitative data about their structure and presumably their function.
Exactly. Quantitative insights.
So here's something for you, the listener, to mull over. Considering these challenges, the complexity of 3D cell arrangements, the need to work with different imaging signals, what kind of powerful possibilities really open up if we can accurately map and measure these hidden internal spaces in 3D? Not just in one plant, but across many different types, maybe even watching them change over time.
Yeah, how might that knowledge fundamentally change how we understand basic plant processes like water use or photosynthesis?
And could it even help us engineer more resilient crops for, changing climate? Thinking even bigger. What other complex, hard-to-quantify 3D biological structures, things currently hidden from easy measurement, could we start to reveal and understand using similar AI-powered 3D analysis approaches?
Lots to think about there.